In this video, I'm going to explain the second part of Lab 1 for microbiology, and in this we're doing a series of sterile transfers. So we are going to be taking a culture from a slant, so as you can see that this is solid, and it's at an angle, the agar's at an angle, so that's what makes it a slant, the agar's at an angle, rather. I say agar funny. Auger is correct. And then we're also going to do transfers from a liquid culture, and we're going to compare them. So we're going to be taking from a slant and transferring it to a broth, another slant, and a deep tube. And then we're going to take the broth and do the same series of transfers. So the broth culture is also going to go into another broth, into a slant, and into a deep tube. And the moral of this story is that it doesn't matter if your culture originated from solid or from liquid, we're going to see the same results. And that's because the cultures that we have are the same. So it's still a cell here, it's still a cell here. They're going to have similar properties no matter where it originates from and where it goes to. So also, because we are transferring these things from two different things, we're going to have to indicate that on our new tubes. So for example, this one's starting from a slant. So I'm going to say that this is slant 2, and this one's a broth. And then I'm going to put the date, whatever the date is. So slant to broth, date, your group identifier, group initials. And that way, and you're going to label all of them similarly to that, like where is it coming from? All three of these are coming from a slant, so you could abbreviate that S. However you understand it, so like this is slant to slant, this is slant to deep. However you understand it is fine. And then all of these are going to be from broth, so maybe this we're going to say broth, and this is another broth, broth to broth. This one is going to be broth to slant, because we're starting with a broth, and this particular medium is a slant. And then this one's a deep. We're starting with a broth. We're going to end up with and we're going to inoculate that into a deep tube. Okay. So the first thing that we want to do is we want to start our sterile technique, and we want to make sure that we have all of our tools ready. So for this lab, we're going to need an inoculation loop that has a long plastic handle. We're going to need an inoculating needle, and we're going to need our media and our starting cultures. Your starting cultures might look different than this. Depends on your instructor's preference. So, and first thing for sterile technique is we're lighting the flame, and we're going to make sure, that it is blue and blue. Blue is the hottest. Blue is going to be the way that we transfer this. So let's clear some of this material out of the way that we don't need, etc., etc. Okay, you can also turn this down just a little bit, goes a long way. And so the point of having the flame is that this is going to circulate the air and the dust up and out because hot air rises. So this is going to keep dust and debris out of our clean work area. So after we have lit the flame, then we wipe down the counter. Lots of people like to wipe down the counter first, but that is not the correct way to do it, because after you wipe down the counter, if you do not have a flame going, things are constantly falling onto your counter. So now I'm using bleach, and it doesn't matter what surface cleaner you use. I like to use one like bleach or ethanol because that does not leave a residue, but you can also use a table cleaner. It's not the cleaner itself, it's more the scrubbing action that gets rid of the bacteria. And then also for good luck, I always scrub off my gloves. This does not make your gloves sterile. This actually doesn't even make your gloves clean. But I like being OCD. Micro is my chance to be as OCD as I want to be. Okay, so now the whole thing is sterile. And I'm just going to show you with one. I'm just going to show you the transfers from the slant. So you can see here it's at the angle onto these, and then we'll do the broth. So first up, and in between each, you want to heat up your loop. You can either heat it from holding the handle from the bottom and making sure, want to tip this up so you can get a better view. You want to make sure that it's orange all the way down the handle, all the way down the wire. The handle's not going to heat up, and you want to make sure that it's red hot. As soon as it's red hot, it's done, and then you got to wait 20 seconds. That you wait 20 seconds to let it cool, because if you put something hot into something alive, it will burn it, and that is not fun. That will destroy the sample. And the point here is to transfer successfully using aseptic technique from here to there. So when you're ready to open, and remember the loop, we only want to touch sample to sample. We don't want to touch the sides. So you're going to unscrew with 2 fingers and pull the cap off. We want to be extra special careful. I'm just going to stick this here so it's not touch. If you have to put it down, make sure it does not touch the table. The table is not sterile. If it touches the table, sterilize your loop again. If at any point you feel it's not sterile, contact your instructor and ask what to do. Most cases, if it's the loop, just sterilize it again. If you need a new sample, check with your instructor. So we want to make sure everything on the outside here is something that we can touch. Everything on the outside is, it might be clean, but actually it's dirty. It's not sterile. It's covered with things. The inside, however, is completely sterile. So this inside part of the cap is sterile. This inside of the tube is sterile. What might have happened, because you see this is the interface where the sterile and the non-sterile meet, kind of. Maybe there's some dust or debris there. So what we want to do is, after we separate them, we want to run it through the flame very quickly. 1, 2, 3. Very quickly, or you will melt the plastic. And you do the same with the cap. 1, 2, 3. Okay, so now this is ready to transfer. So we have our sterile loop. We're holding this with the cap, nothing touching the inside of the cap, because if you touch the inside of the cap, you contaminate it. So none of your fingers touching it, nothing. Oh, the only things that can touch it are things that are sterile. This loop is sterile because we sterilized it in the flame. Our fingers are not sterile. There is no way that any part of our body or our gloves will be sterile. So now we take this loop and you insert it, and you just kind of pull a little bit up. You just want to tap the loop, here, a little closer, onto at least one colony. So now we have a colony on our loop. We're going to close up we're going to reflame our sample. That's hot. Very, very briefly, not melting the plastic. And then we're going to open our first transfer, which is slant to broth. So we are taking it from a slant, putting it in broth, and again, you're going to hold the cap off on the side. This you are not going to sterilize in between, because this has a cell on it. We want to get that cell into here. You're going to insert without touching the sides as much as possible, and you're going to twirl, twirl, twirl with your fingers, just like that. And we have now transferred that cell into this broth. Flame, 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 cap it. And done. And then we're going to do that the same way, slant to slant. So we're going to sterilely transfer from the slant again. So whenever you open it, flame, flame. Did I do that for the slant to broth? I don't know. Whatever. So then we're going to insert after 20 seconds. 20 seconds. We're going to pull a little bit off. Just like you're drawing on Jell-O again, you're just picking up one cell is all it takes, which is kind of creepy because that's all it takes to contaminate a patient as well. And then for the slant, you're going to insert this all the way in. Flame, flame. You're going to insert this all the way in to the very base, and then you're going to make wiggles all the way up, where you're definitely touching the slant the entire way. Then you're going to flame it. And we're going to put this aside, because for the deep tubes, what we need is one of these inoculating loops. So on here, it tells us that these are sterile, so we do not want to reach in with our fingers and grab it. Instead, we have to treat this just as we would any other sterile technique. So up here is like the dirty area, so we can touch up here to open the bag. But then after that part, you want to push push just one all the way up and out, leaving the rest down and in the sterile area. And because we're going to be stabbing with this part, we don't want to touch that part. So we want to keep pushing all the way up until we're pretty comfortable that we're at a spot that we're not going to touch. So here. So we're going to grab this teeny tiny loop end, and we're going to be inoculating with this big fat end that came out first. So you want to make sure that you don't touch that. If you touch it, you've contaminated it. Okay, then same idea. We don't have to sterilize this because it's already sterile. We're going to open our tube and we're going to go flame flame. And then we're going to touch it to one cell. Touch, touch, touch. Make sure. Oh, there, that's it. Touch, touch, touch. Now the cell is on our loop. Cap this back up. Oops, should have flamed it first. Flame, flame. Cap that up. Notice I'm not flaming very long at all. And then for the deep tubes, which are deep agar, if you tried to put this on the loop would bend and you would not get a straight line. So you want to take this and as straight into there as humanly possible, not touching the sides, going straight down. You want to take your time and you want to push it straight down all the way down. And then pull it straight back up. This will not bend. It will be totally fine. Flame, 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 flame. Did I flame that beforehand? I don't know. Okay. And we do the exact same things with the broth. So the broth is a little bit easier to inoculate from. And because this is a demo, I'm just going to show you. Pretend that this is sterile again. First, the trick to the broth is you want to shake it first to make sure that all the bacteria are up and out. Maybe I don't want to use this one. And then you're going to flame, 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 flame. You're going to take this and just dip it straight in. You're just going to dip it straight in, and then, I don't want to contaminate my culture. Sorry, guys. And then, because it's a little bit easier, as soon as it's touched the liquid, it's got the cell on it, and then you just stab it straight in. You would get a fresh one for each one of your cultures. I'm just pretending that that one was sterile. Okay, and the same thing with the loop. You would flame, 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 flame. Make sure that it's out of the flame for like 10-20 seconds. So you wait. And then you would just dip it into the broth. Flame, 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 flame. Waiting 20 seconds feels like a long time, but it's okay. And then just dipping it in. As long as it has touched the liquid, it's totally done. And then I hope you can see that there's a little bit of, you see that like kind of opaqueness right there, like that shininess. That's the liquid. It's now in the loop. So that's where the cells are. Flame, 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 flame. And then to inoculate this into the broth, you just put it in and twirl, and then you would sterilize again, inoculate again, and for the slant, it's the exact same thing. You would just dip it in and drag all the way up with a wiggle, and that is how you do that lab. So we have taken these three cultures, and they originated from a slant. These three cultures, they have originated from a broth. We have labeled them appropriately. We did not leave anything in the flame too long, and now we are done with the lab. So we are going to make sure that our loop is nice and sterile before we return it to whence it came, which is the jar that looks like this. So the next person has a nice clean loop. Then we take our cultures out and we dispose of them properly, which I talk about in a different video, and then we take our cultures from here and we put them in the incubator at the correct temperature for the correct amount of time. Okay, no special storage on this, just make sure that you have where it came from, the date, your group identification numbers, because there's lots of groups who are going to be doing this lab, and it would be a shame if you couldn't identify whose results are yours. Okay, so thank you so much for listening. I hope that this has been helpful.